Good morning, everybody. Welcome to the last day of the summer school, and I see that Barcelona Thursday evening has taken its toll already a little bit and has diminished the public considerably. But still, it's my absolute honor to introduce Jaume Bertranpetit, who is a professor of biology here at the University Pompeu Fabra. And as you can see, he even has a nicer building than we have here as the engineers, because he's directly on the sea. And he is working since long and has a lab in evolutionary biology and complex systems. And what he's doing is trying to model and understand, based on genetics and the genome, how history happened, how humanity happened, and things like that. And he will enlighten us on this. And additionally, it's also partially my honor to introduce him because he's my former boss as one of the ICREA research professors here. So he was long the director of this program and has contributed a lot to the science in general in Barcelona by managing this program, for example. So Jaume, very thanks to coming here in the last minute. We know you're very busy, and we're looking forward to your talk.
Thank you very much, Bart. It's a pleasure to be here, and what I'm going to talk, so I know that you are not interested in the field I'm going to talk about, but I have tried to put the things in such a way of which kind of challenge of information we have and what we ask, so the kind of interrogation we do to the genetic data. And this is the main point I'm going to tackle. So my interrogation is going to be on evolutionary stuff. But you can do whatever you want on that. So I could now switch and say, oh, let's talk only to GWAS studies. That would be exactly the same thing. So to which extent, when interrogating some very, very, very complex data set, that is the genome, can illuminate us in several issues. I'm going to talk on some of these issues, but what I would like to express to you is the kind of challenges we have and to which extent we are able to make the correct interrogation of the data. So as you are going to see, I'm going to put a lot of doubts on many of the ways we interrogate the data, because one of the main points I'm going to tell you is that if we have a very complex data set, and you have that in all the cases you may be working with, the hypothesis on how this data is going to illuminate you is not clear. So how you describe the data, which kind of parameters of summary statistics are going to change for a given conditions, for a given disease, for a given treatment, and which are the specific changes that are going to happen there, and to which extent, when we interrogate this data, we need to have an hypothesis for that or not. So maybe it's not very clear right now, but it's going to be much, sorry. Oops. This is, to me, is the most illuminating phrase I have heard in my life by Sidney Brenner. What is life? Living organisms may be viewed as the only part of the natural world whose members contain internal description of themselves. This first part is fantastic. We have internal descriptions of ourselves. This is why the whole biology must be rooted in the DNA. And our task is still to discover how this DNA sequence arose in evolution and how they are interpreted to build the diversity of the living world. So we have two tasks: how this arose in evolution and how they are interpreted. So these are the points I'm going to talk. The first is an introduction, how the DNA carries the information for the formation of a living being, of a particular species, particular individual. So the challenges of the relationship genotype-phenotype. The second already in evolution, the differences between DNA individuals and species allows us to reconstruct the evolution. So here, as you are going to see, here could be molecular biology. He is going to shift into genetics, and here is going to shift into natural selection. How we may interrogate the genome to understand things on natural selection. So these are the three parts I'm going to talk. The first one, it's a very, very general introduction. I'm not going to talk on that. So how the DNA is packed. Nowadays, I would say that in biology, 1/2 of the people tries to understand the packaging of the DNA, and the other one tries to understand how the DNA is expressed. So nowadays, I would say that these are the two main places. I'm going to talk on the human genome. Just remember the amount of information we have, and in this human genome is going to contain the information to build and maintain the organisms, but also all the non-coding sequences of the DNA. Sequences. The description, the internal description of ourselves. This is what now is what we do every day. So every day we have new data, and nowadays the data is nothing by sequences. But the most important thing is that to have a sequence, a DNA sequence of a given individual, I used to say that it's just a high-tech description of an individual as counting the number of legs of an insect, as the zoologists used to do. So by itself is nothing special. What we have nowadays is that we are able to do sequences because of the drop in prices. This drop in prices, what has produced is an incredible increase, this is logarithmic of course, increase in data. Nowadays we have lots of bioinformatic challenges with that because of the quantity, not because of very special brainware. The problem is problem of hardware because so we are at a given moment that when we produce a sequence, this sequence afterwards is put in some kind of format that doesn't, that is very, very, very compressed. And if you need the previous data, usually you said, well, you have to do the sequence again. It's too expensive to keep all the information that has been produced to get a DNA sequence of an individual. So, for an individual, it's feasible, of course, but when you begin doing that with lots of individuals, lots of information, then it's nearly impossible. So at the very end, there are lots of challenges of getting a right sequence, and I'm not going to talk on that. This takes a lot of effort. And nowadays, I would say that 80% of the people in bioinformatics, they get a job on that whenever you want. So here in Barcelona, people in bioinformatics, there is the BIB, that every week they send around the open positions in this field, and there are every week there are a few. So it's something that it's increasing. The genome. The first thing you have to do is to try to understand the composition of the genome, and the interesting point. There are many interesting points. Here, I want just to stress a couple of things. One of the simple things we always talk is that we all as transgenic, we all as are mutants. I guess you know that you are a mutant and a transgenic. And if not, you can see that here. We can count, we have been able to count the number of novelties each of us have in terms of mutations, in terms of movement of genes within our genome or from outside. At the very end, what we have is just 1.5% of protein coding genes. This is what attires the attention of most of genetics, the attention of most of biology. The introns, which are huge compared to these protein coding genes, and then more than 50, around 50%, so here and here, it's 60% nearly, are DNA that have many copies in the genome, long copies, short copies, retrotransposons. Just for example, we have these 3% of transposons, we have lots of viruses, remains of viruses in our genome, and so on. So, in this structure, we have these two things that Sidney Brennan told us. How this DNA sequence is interpreted and how it evolves. First, how is it interpreted? René Magritte drew this very well-known picture, and the question is, what is he looking here to know that there is a bird? Which is the piece of information? Which is the content of information he is looking at in the egg? For the egg itself, nothing at all. So, in his eyes, there is no information that makes him to make this drawing at all. It could be a stone here, and he could be painting whatever you want. The idea is that he knows from previous experience that from an egg comes a bird. But he's not reading the egg. I don't know whether you see that. I've put here a DNA sequence. This is what we want to do. We, in biology, I've put 2020, maybe I had to put 2030 because it's too optimistic. But the idea is what we want, this first part, is that this is what we want, to read the DNA sequence and draw a bird. This is science. This is really biology. The question, are we there? So how far are we of reading a DNA sequence and saying, this is a bird? What we do in the lab is totally a trick. If you give me a sequence, I would blast this sequence and say, oh, your sequence is a sequence of a bird. I said, how do you know that? Oh, my biology is fantastic. I have expressed this genome in my computer, and the computer says it's gonna be a bird. We do not have this software. This software does not exist in biology today. It will exist, but does not exist. What I do is to blast the sequence. We all blast sequences, but to blast is to do comparative biology, to compare with something that we know, that we extracted this DNA from a bird, and if this DNA matches the one it's in the databases, I know it's a bird. But this is not science. This is just comparing with something that people knew. So we are doing more than Magritte than what we are doing with biology, okay? Because here, what we would need for that is to have a much powerful, a much clear knowledge on the machinery of how the genome is being expressed. And here is, is the point I want to make. So, building the virtual physiological human, we are so far from that. We are so far. Well, but we have to find shortcuts. And because here, to me, the surprise is to see the DNA and the idea is from DNA to the human. So from the DNA, so to put your DNA in the computer saying this is the human that this DNA has the information for. This is what we are for. And I've put here the example in which we know more in biology. This is the mycoplasma. And mycoplasma is the very simple bacteria for which we know even the number of molecules of each RNA, of each protein in the bacteria. And even in this case, we are not able to understand how this information has been generated through the expression of the genome. So the point is that we should have this information ab initio, meaning that what we want is to make the molecular biology work for our purpose. Our purpose may be physiology, may be disease, maybe whatever. But to make that ab initio, in the sense that make the machine work and tell me what happens there. And we are very far from that. And biology has been using a shortcut, and the shortcut has been genetics. Genetics means look at the difference, and if there are differences, I may interpret that. So why biology always is looking at disease? There is just one reason, because that's the easiest way, that's the easiest pathway, that's the easiest case in which biology can work. Because you have something that is working and something that is not working, and you are trying to find the reason for that. Meaning that the biology of the disease is a very necessary field, but this is the easiest part of the biology. So if we do not understand cancer biology, imagine how it will be to try to understand cell growth. Because when cell growth doesn't, or cell division control, because if we do not understand fully division control, cancer won't be able to be understood. And in this sense, we use genetics, you use genetics, because we rely on understanding something with the difference. You are A, you are B. You are A, you are not A. And trying to understand the base for that. So the idea is that biology nowadays is using genetics as much as they can. They are using, and they should be using more, molecular biology to try to build up how this information is being expressed. So at the very end, what molecular biology should, the goal should be to be able to begin a program with the information of a given genome and be able to build a result. And this result could be a mycoplasma, okay? But the shortcut is to go through genetics. Genetics means that we have variation. That's the perfect word. And what I would like to stress here is that variation, it's intrinsic to life, but it's a way of trying to understand life. It's not the way, in the sense that here we rely on variation. And this variation in the genome is what we have to understand how it gets into the phenotype. The idea of this relationship genotype-phenotype is, again, a big part of the biology today, and here we have many different issues of the ways of working in biology today. And when looking at the genome, we may look at all kinds of variation, copy number variants, single nucleotide variants, number, whatever you want. But what has been clearly the most efficient and the most powerful way is looking at just variants of a single nucleotide. Here you have one nucleotide that has a substitution, and here you have another variant. Why this one? Because it has been shown that this is at the base of the most interesting differences existing. Not for all kinds of differences, because, for example, there are many cases of diseases in which is not this the case, but the case is just a stronger disruption. When the stronger disruptions are the ones that are easier to be seen in a genome, and to understand this change in the genome, the phenotypic effects that it's going to have. So I'm going to talk mostly in a SNP, on SNPs, but of course open to any other kind of variations. And what everybody is doing right now is either to look at a set of SNPs or go straight to the DNA sequence. You are going to hear afterwards a talk on how this work, in the sense that these are the way of describing a given individual, and then we are going to pull that to understand the differences among the individuals and how we may make the relationship either with the phenotype or, this will be my second part, evolution. Okay? So the sequencing machines we have right now and the SNP typing, in which you do not interrogate to the whole genome as here, but you interrogate specific positions in the genome. So, for example, this is the I don't remember which one is this. 2.5 million variations in a genome. This is a very standard, the very standard array that is being used. So nowadays that's very, very cheap. So the prices for to have a vision, I don't know, Sistemas Genómicos, what the prices are, but to have an idea, this an array for each of you to know this 2.5 million may cost in the order of 100 euros, a little less, and to have a whole sequence of the 3 billion bases for you is going to cost in the order of 600 euros. So these are the, so when you design a given study, so you weight your budget to see what you may be able to do. Okay? And what is interesting here is the following. We are looking, I was talking on the differences in the genome, but we have some phenotypes to look at. First, we can link the genes and phenotypes and trying to understand why these two individuals are different, or where are the differences between them. So here is when we can make this nice question with a not a very easy answer, which is what makes a human human, in the sense that we should find in just this 1% of differences between a human and a chimp, we should find the differences that are at the base for the phenotypic traits that make us humans and not a chimpanzee. Take care again. We are doing comparative biology. We are not doing molecular biology. I don't take a genome and try to understand this genome. I take this genome, I compare to this one, I look at the two phenotypes and said, oh, here is something interesting. You see the difference in the point. Comparing is a shortcut. Doing genetics is a shortcut for doing biology, because molecular biology is not able to do that, because it's too complicated. And here, what we have is this 1% of differences. And we can do the same question, trying to understand the difference among us. In this case, we know that the differences between any of us is in the order of 1 per 1,000. So of 1,000 nucleotides in our genome, 999 would be the same and 1 would be different, and this is the base, the genetic base of individuality, what is at stake. So I began with the genome. I said that molecular biology would be necessary to understand this information, but immediately I jumped into genetics and comparing sequence with a known phenotype. This phenotype may be disease, not disease, maybe chimpanzee, human, and try to understand the difference in the making of the phenotype. Okay? And now we can begin. I'm going to begin the interrogation of the same kind of data for 2 very different purposes. One is how the differences between DNA of individuals and species allow us to reconstruct the evolution. So my question is, if I have my DNA sequence, I have your DNA sequence, what may I learn from that in evolutionary terms? I'm not going to look for the differences in our individuality. I'm going to try to make questions like, where is the most common ancestor of pieces of our genome? Do we have in our genealogy a common ancestor, and where this common ancestor and when this common ancestor could have lived? These are the kind of questions I'm going to ask here, okay? And here, I don't want you to pay attention to the details. And what I think may be of interest for you is the kind of questions and the way we try to address these questions. So here is where you are going to see the setting of data, our genome, full genome, SNP genome, whatever you want, doesn't matter. Well, it matters. Let's put the whole genome, the information of complete genomes for all of us. How are we going to interrogate this data to try to understand evolutionary issues? This is the question I want to pose to you. And first, I'm going to make some reflections on how can we reconstruct the past in a system that evolves over time. And now, let's make a very idealistic thought on any kind of system, from the evolution of the universe to the most fast evolving systems. Maybe some people would say that is the evolution of viruses, and you can think on the evolution of the language of texts. Doesn't matter. So it may be any kind of system, in this a cultural system, a biological system, a physical system. What do we need if we want to reconstruct the evolution? First, we need a good description of the system, the elements of the system. Second, we need to compare items objectively and quantitatively to be able to make some kind of distances or measure the distance. Okay? For some cases, that's very easy. Knowledge of endurance or perpetuation and transmission pattern over time. So how things change, which is the pace of change in time. And with that, it's possible to estimate the dynamics of the system. The question is, for which system this is known? Is it known for the physical systems, for the evolution of the universe? Is it known, for example, now here, if you look at how biology have been dealing with that till recently, till the advancement of the molecular era, it was a full disaster. Because it's impossible. So you can have a good morphological description of the system, of the biosphere of a given species. But you do not have. So to compare items objectively or quantitatively, here you would have all the literatures of the 80s on the quantitative methods of distances, morphological distance, and all that. But then, when you see the transmission pattern over time, you have no idea. So morphology cannot fit into this kind of setting. And that's one of the difficulties, because this has been tried to be applied to cultural evolution. And in cultural evolution, there are difficulties on this comparison and in this transmission pattern. How is culture transmitted? Okay? But if you have these three, you can go on. And this is what happens with the genetic information, because in genetic information, we have very good description of the system, the sequence. We can compare objectively and quantitatively, comparison among sequences, and we know the transmission pattern over time and the mutation rate. As we know the mutation rate, if we have 10 differences and one difference happens every 10,000 years, our common ancestors lived 60,000 years ago. That's it. As simple as that. So we have to model that in some way that really works. And this kind of thing, it's very powerful. And when you can use that either to work with the whole diversity of life in the top, or go to a small fraction here and try to understand the differences among mammals, whatever you want, or the difference between individuals, the difference wherever you want, or to apply that to other systems. 2 years ago, when I left ICRE, I went to Cambridge for a sabbatical, and in one of these college high tables, it happened to me that I seated at the side of an astrophysicist, Paul Geoffrey, and we began the conversation that they were classifying stars. I said, but what kind of old-fashioned things are you doing? Classifying stars. That was done in biology by Linné more than 2 centuries ago. We do not classify. We do phylogenies. We do not cluster only. We do a clustering that may be read in evolutionary terms. And we published this paper of cosmic phylogeny, of which I understand only the first pages, of course. The remaining I don't understand. But the idea is the same. So the main idea of the paper is how it is this system of stars, so here we have the sun, how the system of the stars, according to the decomposition of elements, may be understood in evolutionary terms, in phylogenetic, not in classification by itself. And this is the interesting point. So what we have is that a given moment here, right now, past time here, and what we have is what we may observe in these points, and the question is, may we go back in time with that? And this is what we have tried to do in evolutionary biology, or evolutionary genomics, if you want to call it that way. And for that, we need to dig really to see how the information is hidden in the genome. It's not always obvious. If I show you this picture, you said, okay, I understand what it is, but maybe what you want to see is something like that. So how can you see, can you recognize that this bee is going to give this pattern in the shadow of the water? And this is the way we are going. I'm going to show you how we analyze this information. So I'm not interested, or I guess you are not interested in the specific ways we do that, but it's just to give you an overview of this kind of things. So for example, you have read in the newspapers that we have an African recent origin. How do we know that? Well, this is phylogeny, just as simple as that. If you give me several sequences, it's something not difficult to know. I'm going to show you something on that. So genome data supports a single out-of-Africa migration event for all populations in the world. So it's just a way of trying to disentangle these two hypotheses. One hypothesis is that in this out of Africa there is one initial wave that went into South Asia and the Pacific, and another that went into Asia, but another possibility is that everything comes from a single one, meaning that the populations here have the same time depth, in this case here, the same time depth, as the other ones of the diaspora out of Africa. And if we are able to make a tree of these populations in which we know the pace of this tree, we are going to see the following. So this is just what we get with whole genome sequences. Lots, it's crazy the amount of data that is there, but the most important thing is this is the most, the closest tree in which the branches have a proportionality to time, okay? Because this element here is an element of drift, we call drift, and which is the random variation that is being produced by the random inheritance of the genomes. And in this case, what we have is a given structure of populations, and here what you have, the initial one is that you have the ancestor, here would be the chimp, or all the other species, and this is the human branch. And in the human branch, first, what you get is all the African populations and the remaining populations. Okay, let's see that in more detail. So we get into the Africa and non-Africa. With as simple as that, you have this idea of the out of Africa. That's it. You don't need more information, because here you have the whole human genome, or the diversity of the human genome. Here you have all the non-Africans. How is it structured out of Africa, this variation? You have the Europeans by one side, you have the East Asians by the other, and here you see that Europeans are an outgroup of all Asian populations, and next split is the Pacific populations here, Australia, Papua New Guinea. What does it mean? Means that there is a single out of Africa, one branch gets into Europe, the other branch goes into Asia. There are no two waves into Asia. A single one explains everything, and the amount of diversity of these individuals is similar among these three main branches, which is something that for many years was not accepted at all, because in Southeast Asia you have these negritos that were called the pygmies, that are very different from others, and the morphology had totally mistaken anthropologists with that. So the idea is that as we get something that the pace changes with time, we are able to recognize that. The second example is more complicated, and I'm going to try to show you why and how is this idea you have also read in the newspapers, by which we have a little bit of Neanderthal genome in our genome, okay? So we, all of you know that we have some Neanderthal genome. The question is, how in the hell do we know that? Which is the kind of analysis of a genome sequence that may tell us that. This is what I am going to show you. So, and I'm going to show you that we have not only Neanderthal, but also there are populations having a little bit of another one called Denisova, and we have also, again, proposed that there is a third component we call unknown hominid ancestry. So the idea is how this information of the genome may tell us this kind of details. This is the general setting. So Neanderthal is here. This is the branch of Neanderthal that went extinct some 40,000 years ago. And we have a common ancestor, of course, Neanderthals, Homo sapiens. Homo sapiens, we have already seen that, the out of Africa, then the Africans in Africa, then the out of Africa, which is here, that giving rise to all the non-African populations. And here you have this Neanderthal introgression, okay? Which is the kind of interrogation of a DNA sequence that may tell us that. I'm going to begin with something interesting, is that for some 10 years ago, it was possible to obtain a DNA sequence, a whole genome sequence of Neanderthal. This, the world of ancient DNA is fantastic, and it's always surprising because they are able to get genome sequences of incredible, complex, extinct things, and meaning that we have a genome to compare. So the idea then is, let's compare this genome to our genome, and let's see if we can get the pieces of our genome that comes from this Neanderthal. And the idea is very simple. You can take that as a specific position in the genome, and in this specific position of the genome, so here is the diversification tree. This is the external node in which you have A is for ancestral, D, and D is derived. I mean, the change is from A into D. Okay, this is the ancestral, and it derives here. That's the reason why you have here derived. That's the reason you have here derived. But here, look at that. There are cases in which you would expect that if this is ancestral, this is derived, this is going to be derived and derived. Okay, this is that would be normal, because the mutation would have occurred in this part of the tree. But we have a surprise. And the surprise is that here is an A, here is an A, but there are a D in one or the other. So the question then is how we can interpret that. If we have more of one or the other, it's just to see whether this kind of tree is more frequent than this kind of tree. You see, it's just to show you the kind of trick our mind has to do to make this kind of interrogation. If you have a negative score, it means that these two are more similar than expected, and then it means that the tree is not like that as it was, but is like that, or that you have admixture, introgression between these two branches. Okay, so it means that this D has come here as a D. This is the kind of setting that allows us to see when you know that this is not, so here you see the case, and I mean, oops, oh, sorry, I was in negative score. You have French and Spanish, of course, this is the case, and that's very simple. But if you have a Neanderthal that we know is extinct and happens that, means that there is admixture between these two. You understand the point? So there are more amount of derived of cases in which we Europeans are similar to the Neanderthal than to Africans. The idea then is that it's possible to recognize with the Neanderthal that all the non-African populations have this excess, but not the African. And this is the way of recognizing that from this population there has been a certain amount of introgression that now we calculate in the order of the 2%. Okay? The idea, again, is how this kind of questions may be solved digging into the data. Years later, the group of Svante Pääbo published a paper in which they sequenced what is called the Denisova remains. The Denisova remains is a piece of bone like that, and they got the whole genome sequence. We don't know how it was. It's the only remain. We don't have a skull, we don't have nothing else. This remain, and we have a DNA sequence of something that we don't know how it was, nothing else. There is a tooth and the remains of this fragment. And this Denisova was found in Central Asia, and doing this kind of analysis, we see 0, 0, 0, 0, 0, 0, oops, here, we see that there is introgression in Papua. Doesn't matter. I'm not going to discuss the evolutionary issues, but just to give you a flavor of the kind of interrogation of the sequence data we can do to recognize that in this population there is this amount of sequence that comes from that, because there is no other way to explain if this individual was extinct again, like the Neanderthal 40,000 years ago. Okay? And to go a little more in depth with that, we have been able to show that it's possible with the same technique to find that there is also in some populations a certain amount of another hominin that is not neither the Neanderthal nor the Denisova, and in this case the analysis is a little more complicated, but at the very end there is an excess of highly divergent sequences in our DNA. So it's not possible to understand how this amount of strong divergence may have been built if the evolution of our genome has not been adding a third component, which is here. So here what you have is modern humans here, Africa, non-Africa, and here you have this Neanderthal and Denisova, okay? But we have found a third one that is more divergence also in this genome, being able to postulate that this admixture is not only Neanderthal or Denisova, but there is a third unknown Southeast Asian. So, but the idea then is how the DNA data may be interrogated to get all that. And in these cases, the point is that we need someone to have think on this kind of statistics I've shown, this D statistic, to be able to disentangle that. But every time more and more, we think that there are many issues in this DNA structure that are hidden in the complexity, and maybe our intelligence is not enough to be able to recognize which are the past events that gave rise to that. The idea is, to which extent are we able to pinpoint the effects of the genome of the various demographic events? Are we smart enough to design statistical tests for each hypothesis we want to test? How to use simulation and an open frame to estimate parameters that may affect the genetic structure? And here is just to propose you that we may need a much more in-depth setting to do this kind of evolutionary analysis. So we have DNA and we want to have a model and to estimate parameters. How can we get into that? And it's beginning, so what I'm going to show you now is just a beginning. We are not very sure that is going to be the right path at all, but is a path that is extremely interesting, that going to, this is the standard ABC analysis that I'm sure you have seen in many other settings, in which we do simulations, we estimate from the simulation summary statistics, we compare the summary statistics of the simulation with the observed, and we retain the simulation parameters given a threshold. So this is the standard ABC analysis. But there are several problems that may be solved if we are able to put here a very complex way, a very complex setting of the sequence data in order to be able to get into a deep learning setting to be, so the idea is we don't know how the summary statistics are being built inside to give us the making of the parameters we are trying to calculate. As we don't know that, we leave these deep learning algorithms to get into that, because we know that many of the functions, all the functions are not linear, and there are lots of interactions among them, so we don't know how to read the DNA sequence to understand, for example, how many people lived in a given population, but we know that the information is there. And we leave this deep learning setting to do that, and we can ask, if we have all these models, here are all the possible models of introgressions and human evolution, whatever you want, the question is, if now I put, these simulations, these models into this setting, which are the ones that fit better the data, and then you get that some of them, they have a very extreme good likelihood and others have zero, and at the very end we think we are getting into a model in which we have the integration of this Neanderthal, as I showed you before, of this Denisova, and this third hominin, for which now we have been able to put exactly. We think that we have been able to recognize when this population lived and the phylogenetic position. And here you can see that it seems that from the original distribution of a given parameter, in this case it is a population size of one of the populations, we are able to improve a lot our calculations. So these three examples I've put is not to tell you that these are specific analyses that are of interest, but that there are many ways of analyzing this genomic data for which we could have this information, and ways of interrogating data that really may tell us something that is at all is not evident. But knowing that the information is there, there are ways of getting. And this will be applied. I'm sure that in the next year, so till now in biology there are, in biology, not in face recognition, deep learning applied to biology, there are 2-3 papers, not more than that. Next year is going to be at least in the hundreds. I hope that our will be among them, but still is not there. And just to finish, I'm going to show you the same thing, but now how, again, these differences in DNA allow us to infer where natural selection acts. So it's just a new example of this idea of understanding how we can interrogate this genomic data now for another different thing, for something that I'm sure most people have not think about. The idea is that we can revisit Darwin in the sense that natural selection is at the base of the diversification of all the species, the diversification among populations, and that the diversity that we can see, this diversity of the finches in the Galapagos, the Cypaena moralis differences according to the selections, if the birds, all these cases, can we revisit that with the genomic data? And here I'm going to show you just a couple of points, and the idea is the following. If this is past and present, and let's look at this example here, we have an amount of variation, the variation existing in this room now, right now, and imagine that at a given moment one of these variants here, this one, is being selected. So this is this gives a higher fitness. So with time, this is going to increase frequency. At a given moment, is going to be fixed. There are lots of things for which in our genome we are the same, 99.9%, meaning that all these are fixed. The idea is, if now we look at these individuals, we here, and look at the genome, how are we going to infer what happened in the past? The idea is very simple. If you look at these genomes, you find an amount of variation. If you look at this genome, there is no variation. I need not just one sequence of a single individual, I need the whole population to realize that here in this position there is a decrease in the amount of variation. That's it. So I do this, the genome sequence for all of us, and I put these genome sequences one with the others, okay? Align it. And then I ask, which is the amount of differences among us, and you are going to see along the genome something like that, and suddenly, and here there is this decrease, means there is selection has been acted in the past. And this is just to show you how this may be interesting, because it can interrogate the genome for something that happened in the past. And we have several ways of interrogating the genome. I'm not going to talk on that. And some of the footprints are very old, some of the footprints are very recent, and some of the footprints may be acting right now. So to which extent natural selection is acting on our genome today? We all think that if it is acting, it's tiny, the action of natural selection, but we may ask, for example, when people, some here, some in Europe, some 8,000 years ago, began producing food, to which extent natural selection changed? Our food changed a lot, our way of life changed a lot. So we can do this kind of questions, and here you have some of the cases we have been able to work out in the human genome. Some of these are very nice and interesting. So for example, in North Europe, people do the most incredible thing in nature is that when they are adults, they still drink milk. That's crazy, guys. To drink milk as an adult, that's biologically, What happened here? So it was a mutation of producing lactase in the adult that happened to be very successful, because in that population in North Europe, around here, they already had dairy food. They had cows. So these people got an extremely good mutation, being able to digest the milk. And we have this, but this is a genetic variant driven by natural selection. And this is the kind of things that have been able to be found in the genome. Resistance to malaria, there are many different ones. We have, for example, resistance to cold. The most interesting is adaptation to high altitude, which are here, here, and here, so and so on. The main idea is that in all the cases this is feasible, to be recognized that natural selection acted in these places, and among the ways we may interrogate the genome for that would be the following. I'm not going to talk on that because it's going to be too long. Now it's time to begin finishing, and I'm going just to talk on one of these. So we have to be sure that the methods are robust and good. We may be able to understand polygenic adaptation that is very complex. So for example, adaptation for skin pigmentation, adaptation for height, for body size, and there are very interesting cases of that. One of the questions that are still open is to which extent we can detect differences among human populations on adaptation for psychological traits, for example. Cases in which we have this integration. So for example, the question is, the entrance of some Neanderthal genes in our genome was good for our adaptation. Some of them were used for a new adaptation. And I'm just going to show you very briefly this whole genome selection analysis, because this is very similar to the GWAS analysis that I'm sure it's frequent in your case, and that you have seen many more in. Just a second, I'm going to just to point to that, and because yes, I have to leave time. And here you have the case of a migration of a population from India to Europe. These are the gypsies. And when comparing this population, Indians, with the population that migrated with the population that lived here, we have a very general setting of changing environment, and look which are the adaptations that took place. And this is the kind of analysis we do. So here, this is a kind of Manhattan plot, very similar to the GWAS analysis, in which people detect genes for complex traits. Have a look. It seems that today in Science there is an interesting paper with new data for psychiatric diseases. I haven't seen it, but it seems that is quite interesting. And here what you have the 0 here, and you have a test in the top, another one in the bottom, and then the idea is very simple. When you have that means that selection has acted, okay? And here you have the whole genome, chromosome 1, chromosome 2, chromosome 3, 4, 5, 22. And then we have to go into a given place and to say which genes are there in this region that may be important, may have been selected. And I'm going just to show you one of these cases in which we have the data. Oh, I thought, sorry. In this case, I'm going just to show you that, we have been able to show that one of these big ones is related to pigmentation. Pigmentation is something that has been changed very recently among humans, and here you have the toll-like receptors that we were able to prove that was related to the Black Death in Europe and the resistance to the disease. The idea then, to wrap it up, is the following. The genome, the way that genome expresses itself, the way that genome changes in time and in space in this single evolutionary setting. Differences among that may be read in many terms, in terms of health and disease, in terms of evolution, population history, and in terms of evolutionary adaptation or detecting natural selection. Thank you for your attention.
Thank you very much for this very interesting and thought-provoking lecture. Any questions? Let me maybe start a discussion with what you started with at some point, the difference between kind of genetics and biology.
Yes.
So you say that what we might have, you say 2020, maybe later, is that we can start from a DNA and then kind of simulate, let's say it that way, the organism that would come out of it. Now, there was, if I remember well, was a talk by Dennis Noble, which is one of the popes in simulations. And he said that if you would start from this...
The physiologist in Oxford, you mean?
Exactly, yeah. And he's like a bit of one of the fathers of this VPH and the physiome and the whole kind of computational modeling of the human body. And if I remember well, in one of his talks, what he said is like, okay, say that we would start from the DNA and then try to simulate. So make a simulator that would do this. Because of the complexity, all the interactions, all the proteins, all the environmental things, you would end up with a computer which is a human. Because the only way to simulate it is make the organism in some ways that can simulate it. So the type of computer that you would need is more or less the body. So then you can say like, okay, maybe we just study the body and think differently about simulations. So I don't know what your opinion is, whether you have been thinking about it, like which are the things which are useful and which are needed and which can really address questions when we go towards the more like the modeling side, for example.
So what I see is that you have heard Professor Noble several years ago, not in the last 2-3 years, in which
No, exactly.
All his talks are bullshit. Bullshit, okay? Am I clear?
Very much. And it's a phrase I use very often, I have to admit. So we have a lot in common.
So a good physiologist, a day became, he wanted to become a kind of Dawkins selling books of popular science, and he wrote one of the most disgusting books I have seen in my life, in which he makes lots of invention of epigenetics in evolution. So you have not talking on that, and we leave that aside. Okay, so don't read this book because it's pure bullshit. The idea of the complexity, that's very nice, the idea you have said, Bart, in the sense that it's so complex, but we want to do everything in silico. You want to do everything in silico. We have to be able to do that. And in a certain way, I mean, people are trying to do the brain in silico, which is much more complicated than many other things. So there is no reason of being able to do that. And if we have to put noise, we put noise in our systems. If you have to make complex interactions, we do the complex interactions. I think we should be able to do that. We should. I mean, it's a challenge. We want to do that. I mean, there is no reason to say we are happy and that's it. No, it's not enough. So it's true that many things are very complex, but at the same time, I mean, all the work you are doing is very recent, the tools you have. So for how many years have you been able to use computers powerful enough for what you want? Nothing. Every day you fight to get more powerful computers, and we are going to have them. So my view is that yes, we have to fight to get into that, and my feeling is that we will need, and this is what you know much better than I do, is to go into this kind of neural networks, deep learning, because the difficulty is many times is our own way of thinking. As a professor I had many years ago used to say, we have always to think that our brain came to survive, not to understand the world. So we have difficulties on that. We are very good in surviving, but not so good in trying to understand the world. So maybe what we need is a new brainware from the computers.
Just to continue on this, the phrase that I've been using about deep learning is that I think it's bullshit. So we have from the other way around. So a bit talking about this, that you said like, okay, I'm trying to do deep learning for this kind of things. It's like one of the things, well, there's two things with deep learning. Is first of all, you need to have data, annotated data. Which is the annotated data that you use? Do you have ground truth in some ways?
So the idea, what we do is the following. We have simulators in which you put, you have DNA sequences, you generate, and then you grow that in a given demography. We put demography, only demography. And then, so we said, for example, we are going to put a big population size and a split and a migration. And then another thing, and then,
so you learn on simulated data?
Yes.
Okay,
yes.
Exactly.
Yes.
And one of the other things that deep learning does, it actually smooths out rare occurrences. That's a typical thing that you start to see. And so the question is, how important are these rare occurrences for your type of analysis?
We don't know, of course. Your point is that we don't know. Always the rare occurrences is what it's always impossible to pinpoint, and which is always the place we said, oh, maybe this is at the base of what is missing in our model. We don't know at all.
Question there?
Hi. I don't mean to be very controversial, but I am a big fan of machine learning and deep learning in particular. So just like, it is a misconception that deep learning just is like an average behavior. That's not true. In fact, in natural language processing, what you do is actually you try to predict the rare words in sentences, and similar approaches are done in genetics. And my question to you was actually about, because traditionally deep learning is used as a predictive model. So you try to learn from the data so that you can sort of see what's the outcome, but you're trying to use it the other way around. So you're using deep learning to not to predict, but to see what was before. So my question is, how do you feed the data? How do you train the models? Because it seems like a bit an unorthodox way. And I saw that you use some summary statistics and so on. So it intrigues me a bit how the model learns to see what's in the past as opposed to see what's going to come.
I think the best one would be, I'll send you the manuscript. Okay?
Okay.
It's under second revision, I guess. It will be accepted.
Okay. And a bit less technical. Do you see a place for machine learning in these kinds of studies?
A lot. For example, I have not explained it to you, but for selection, we are also using that a lot. I mean, we need that because, I mean, the amount of complexity we get, for example, if for selection, we have 30 different people keep publishing ways of detecting selection. So what in the hell are we going to do with all these ways in which selection sees something in the data, this something else, this something else, this something else? Our brain cannot handle 30 tests, selection tests in 3 billion base pairs. We cannot handle that. And we need that.
But still, how do you summarize, like if you want to do a genome-wide study, how do you summarize it to feed it to a deep learning algorithm?
Oh, no. So for the case, it's very different for the selection in which you want to see all along the genome, okay? And in the case of summarizing, the question of summarizing it is a question of which summary statistic you use for the description of the data, okay?
Yeah,
Strong fight on that, and I think we have tried to solve at least we have focused a lot on that. If you give me your email, I'll send it.
Yeah, of course. Thank you.
Yeah, I have a question. So first of all, thank you very much for your talk. I'm a fan of National Geographic documentaries, and I could have perfectly listened to your talk in my couch with a beer. So, I would have felt fantastic. I have a question. Maybe that's a stupid question, but if you just have a piece of tissue that comes from very ancient times, how sure are you that you have the complete genomes? Because morphologically speaking, maybe less than 2% of genome variation can make a huge difference, making the difference between monkeys and humans, for example.
Yes. The challenge is huge, as you know very well. So there are ways. For example, nowadays, one of the most interesting things now so. For example, if you have ancient DNA, it's going to be a little bit degraded, and there are some changes that happen, and these changes happen at the ends, at the extremes of the molecule, not at the center. So now, if you look at any paper of ancient DNA, you are going to see a graph which is very strange, and which is something like that and something like that at the extremes. And they say, as you can see, there is degradation in the extremes, meaning that this is ancient DNA. So there are ways of always this discussion is going to be endless. But because the more different, the more sure you are that is ancient DNA, meaning that you may bias that into the most different and ancient part, thinking that what is more close to us is going to be deleted from your database. And this has happened. This happened some years ago, and now the techniques begin to be so refined that even in a work someone in our lab did a couple of years ago, no more, 4 years ago, he was able to see even the amount, so the DNA of the archaeologists that had handled the bone, and the degradation with the 20 years since they had done the study of the bones, the degradation of the DNA of the archaeologists during these 20 years. So people in ancient DNA are extremely good nowadays. Extremely good. I mean, sometimes they do things. So, for example, now it's the fashion is not to try to get the DNA from a bone. It's just to get from the sediment. You extract the DNA and you find everything, all the animals and everybody that pee there.
So I guess I will pass the mic there, but on the way I have another question. So I guess that you have like uncertainty propagation models, because when you want to make the inference between a SNPs or genetic variation in a specific function, which is a set of SNPs, sometimes you're not sure that you have the full set of genetics that would explain a change of function or an adaptation that you're interpreting. So I guess here you should have, along all the machine learning algorithms, some models of uncertainty propagation to give some...
You mean to obtain the sequence or to recognize that the adaptation is there?
To recognize that the adaptation you identify from the sequence is related to a certain functional adaptation of the population.
This is not straightforward. So the final relationship phenotype-genotype, many cases you have to go to the wet lab to make to put the mutations in your system and see the phenotype. In many cases. So we consider that it is really demonstrated if you do that going back to the wet lab. Yes, unfortunately, but this is the case.
Thanks a lot for the talk. I'd like to go back to the picture when you showed like the egg and the bird, and that the final aim would be to have a genome and then kind of simulate and find out how the human being will be look like. And
let's put a bacteria.
Okay, bacteria. And but what's about the other side of the medal, like epigenetics, for example? That papers show that the 50% of variation or 50% of information is due to epigenetics and not the genome itself. And I mean, if we consider humans, their environment is so diverse, and every human is kind of living in different environments. If you take population, it could be maybe averaged, but if we will model the human, like for this particular genome, it's so many different environmental factors. How we will deal with it?
In the last years, and this is the point that Nobel took, there has been a misunderstanding of the problem of epigenetics. Epigenetics, unfortunately, it's a kind of new wave, epigenetics. So modern people now, people in non-standard medicine, they talk on epigenetics and they don't know what it is about. But it's a kind of new wave thinking. The idea of epigenetics is that the regulation of the genome is essential, and this regulation has many ways of modulating, mainly modulating the chromatin, and this may change independently of the DNA sequence. How it changes? The main changes we have are changes that, in a certain way, are induced by the information itself of the genome, in the sense that what is important for epigenetics is how epigenetics works with development. In you and me right now, epigenetics is not that important, in the sense that we have. It's important because in our cell of our body, 90-something% of the genes are not active and the others are active. But the way that activation, deactivation of genes work, gene regulation, we have to talk on genome regulation and not on epigenetics, because genome regulation is the key feature, using epigenetics and using other things. But the idea of the genome regulation is something that the genome knows how to do it. So at the very end, when your mother was pregnant, she knew that was going to have a human and her own son, meaning that every cell knew very well what to do. So the regulation was very well established. You said, oh yes, it may be something that could change because in the egg there was that given protein. Yes, it may be important that. But the world of regulation is very well regulated. So now this new wave thinking by Nobel and these people is like refinding the freedom in life thanks to the epigenomics. I said, what in the hell? My cells are not free. They had to make a human. They were not free to make a C. elegans. No. So to me, in the last years, there has been this increase in the new way of thinking that life must be free, and everything may change. So I have to concentrate myself because I'm going to make an epigenetic change. It's like that, guys. It's like that. It's like that. And this is not clear. I mean, the genome regulation is extremely good. It cannot play games. Games become cancers, most of them. So there are changes that are due to environmental factors. Of course there are. But in the mainstream of making a body, this is something, this is a kind of freedom that life cannot support.
I think that's a very interesting phrase to end and go to coffee. Once again, thank you very, very much for the talk and especially for the discussion.
Thank you, Lars.